Hello, welcome to the fourth lecture of the cardiovascular section of the Integrative Systems Physiology course. Today we'll be focusing on cardiomyocyte contraction and the cellular mechanisms contributing to that event. So during the last lecture, we predominantly spoke about action potentials in the heart, and how those are subsequently translated into ECG signals that we can use as a diagnostic tool to understand some of the physiological changes that occur both over the course of the cardiac cycle as well as in health and disease. Of course, there's a lot of focus on cells that have automaticity. But today, what we're really going to focus on is the cells that actually don't have automaticity, and that wasn't the focus of the lecture last time. And so what's interesting when what was observed by a number of students during our discussion was that the morphology and duration of the action potential in these non-excitable cells is very different than what we observe in cells that have automaticity. And as we discussed very briefly, this is due to a component of the action potential phase where there's substantial calcium influx. And so again, when we're thinking of how we would design action potentials to drive physiological changes that are important for the function of the organ, that is where we start to think more carefully, certainly within the cardiac system, about calcium influx and the role that will play in cardiomyocyte contraction. So if we look at this with a bit more detail as compared to what we did last time, we've got this action potential of a cardiomyocyte, which could either be atrial muscle or ventricular muscle, and that's depicted here in this figure. And this is broken down into 5 phases. Of course, they're starting with phase 0. And so you can see here in phase 0 what we have, and we're looking at the electrochemical gradient. You can see that at the initial very rapid upslope here of this cardiomyocyte action potential, we have a very robust influx of sodium during this phase, which is largely driven by the opening of sodium channels once the threshold potential is reached, and a very large electrochemical gradient, which is driving sodium into the cell. During the early phases here of this depolarization phase, then we subsequently have the activation of rectifier potassium channels, which allow for the efflux of potassium from the cell. Again, now we've got an electrochemical gradient that's favoring the efflux of potassium out of the cell. That results in a very slight repolarization here. But then before this repolarization becomes too substantial, what we have is the substantial influx of calcium into the cell through the opening of these calcium channels. And so again, now we have a very sustained, long duration action potential, with this largely being driven by the voltage sensitive calcium channels. And then ultimately, as those channels close, we have the repolarization of the cell, largely again due to the efflux of potassium, and then ultimately returning back to baseline here. And so those are the 5 phases of the cardiomyocyte action potential that I think it's important for you to take into consideration and appreciate and understand. So, again, I think you could say, well, that's totally fascinating, right? But at the core of what we're trying to get to today, the big question is, how does this actually result in cardiomyocyte contraction? And in the core mechanism and the key event that occurs concomitant with this depolarization and the influx of calcium into the cell is a process called calcium-induced calcium release. And that is pivotal for having sufficient calcium within the cell to elicit molecular events that enable contraction. And at the core of it, again, the increases in intracellular calcium are what enable cross-bridge cycling. And so when we start to think about how are we going to get to a point where we have sufficient calcium to elicit this cross-bridge cycling event, what we need to have is a process called calcium-induced calcium release. And again, here, the issue at hand is that it's very inefficient to have all of the calcium required for this process to come from an extracellular source to an intracellular location and then extrude all of that calcium over and over again. And so with calcium-induced calcium release, what happens is that these action potentials open up voltage-gated calcium channels. You can see that here in these T-tubules, or these invaginations within at the points of the Z lines usually, where the calcium channels are activated by an action potential, and that influx in calcium is an initial event with the opening of those voltage-gated calcium channels. Now, after that calcium gets into the cell, that elicits the opening of ryanodine receptors on the sarcoplasmic reticulum. And so that is shown here. You can see calcium influx into the cell, calcium binding to the ryanodine receptor on the sarcoplasmic reticulum. And that results in a very large increase in intracellular calcium into the cytoplasm. And that by itself is calcium-induced calcium release. And that release of calcium from the sarcoplasmic reticulum is the primary mechanism for an increase in intracellular calcium during the cardiac cycle. Now, once that calcium is released into the cytoplasm, the calcium binds to troponin C, which we'll talk about more in a minute, and ultimately that enables a series of events that allows for cardiomyocyte contraction. And then at the cessation of that action potential, when we start to have the repolarization of the membrane, we subsequently have the closing of the voltage-gated calcium channels, substantial calcium reuptake into the sarcoplasmic reticulum. That's done via an ATP-dependent mechanism here, as depicted in the figure, as well as a more minor mechanism of extrusion through pumps out of the cell. And so here you can see for 5A, which is ATP-dependent reuptake of calcium into the sarcoplasmic reticulum, and 5C, which is this sodium-calcium exchanger. You can see that we can bring sodium back into the cell, and we can extrude calcium out of the cell as major contributors to clearance of cytosolic calcium and enabling cardiomyocyte relaxation. Minor contributors of the cell of extrusion of calcium from the cell include ATP-dependent mechanisms for the removal of calcium. So that is, in a nutshell, the mechanisms that are contributing to calcium-induced calcium release. And so there, you say, well, that's great. We know that in response to us, when under normal conditions we just sort of hang out, and that works very well. Of course, in response to a stress, we do have to have modulators of calcium-induced calcium release, because that is going to enable increases in force production and increases in cardiac output and stroke volume that allow us to meet the metabolic demands of the system. And so what are some mechanisms that are primary modulators of calcium-induced calcium release? And one such example of a modulator would be catecholamines, such as epinephrine and norepinephrine. And so these are a part of the fight or flight response. So you really want to have an increase in cardiomyocyte contractility or force production and enable an increase in cardiac output. And so when we look at how catecholamines increase force production in the myocardium, it is through the augmentation of this mechanism of calcium-induced calcium release. And so what you can see here is that norepinephrine, for example, would bind to a beta-adrenergic receptor, which is a 7-transmembrane G protein coupled receptor, that would result in increases in intracellular cAMP or cyclic AMP. That would ultimately increase CMP kinase and result in the phosphorylate increases in that kinase, which phosphorylates proteins. One such target is a protein called phospholamban. When phospholamban is phosphorylated, that ultimately increases the activity of this ATP-dependent calcium pump in the sarcoplasmic reticulum. So if you think back to what occurs in the preceding slides, during that diastolic phase, where normally you've got this balance of ATP-dependent extrusion of calcium from the cell, now instead of relying on that and as well as just the quote normal levels of calcium uptake into the sarcoplasmic reticulum, what you'll be doing is dramatically augmenting the amount of calcium that is taken up into the sarcoplasmic reticulum during that subsequent phase of relaxation. So during the ensuing cardiac cycles, where now, again, you have calcium binding to the ryanodine receptor, and that is resulting in the opening and release of calcium into the cytoplasm. During those subsequent beats, you will have a larger release of calcium into the cytoplasm under conditions of catecholamine stimulation, and that will result in higher levels of cytoplasmic calcium and subsequent increases in binding to troponin C and force production by the myofilaments. So again, just as appreciation of a major mechanism that increases calcium-induced calcium release, catecholamines would be one. And again, you can say, well, this is fascinating. It sounds like a great compensatory mechanism. Well, what happens with chronic beta-adrenergic receptor activation? Because this seems like a great mechanism to augment cardiac output under conditions of chronic heart failure, for example. And the challenge here is that actually nothing really good happens with a very chronic beta-adrenergic receptor activation by catecholamines. So two things happen. One is you have receptor downregulation or desensitization, very well established. And so there you start to lose your ability to increase contractility via this mechanism through receptor desensitization and downregulation. The other is that you actually have cardiomyocyte apoptosis. So not only when you get activation of the receptor, you're getting poor activation, you're also having apoptosis of some of the cardiomyocytes, which ultimately decreases your total functional number of cardiomyocytes and impairs cardiac function. So, and interestingly, it's not just a theoretical, animal-based experiment that people would say, well, this this could potentially be bad for patients. It's actually been shown empirically that beta-adrenergic receptor agonists or stimulators, they increase mortality in chronic heart failure. And so there was a drug called Zemoterol that was tested in the 1980s as an agonist. It seemed like to be a very intuitive thing to do. Let's increase contractility and we'll improve outcomes in patients with heart failure via stimulation of the beta-adrenergic receptor. Mortality was increased in these patients, and so it's empirically been shown to be deleterious as a therapeutic target in these patients. So that broaches the question, well, what can you actually do to increase contractility pharmacologically? Because it is a tremendous clinical problem of having a heart failure and a huge expense and burden on our medical system. And one mechanism that can work is through the administration of cardiac glycosides. And so here, what you have is the inhibition of this sodium-potassium pump by cardiac glycosides like digoxin or digitalis. And there, what that does is that reduces the extrusion of calcium out of the cell. And by reducing the extrusion of calcium out of the cell, that again, by a somewhat similar mechanisms, enables higher cytoplasmic calcium levels during subsequent cardiac contraction cycles. So again, we're looking at something that is going to increase force. Now, unfortunately, much like the observations in patients that have received beta agonists, in terms of very long-term treatment with cardiac glycosides, generally there are increases in mortality with these patients as well. So again, it hasn't proven to be a highly successful mechanism for improving outcomes in the long term, although there are short-term advantages to dealing with acute decompensations of these patients. So, now we're at a point where we say, well, how do increases in intracellular calcium per se, how do those result in increases in cardiomyocyte contraction? Because we've kind of talked about everything up until this point of the action potential and how that action potential increases cytoplasmic calcium, and then it does something downstream. And so what is that downstream molecular event that enables cardiomyocyte contraction and shortening of those cardiomyocytes to ultimately eject blood into the circulation? And so that is done down here by the sort of workhorses of the cell, which would be the myofilaments. And so within these myofilaments, what we have are thick and thin myofilaments in cardiac muscle. And so the thin filament is comprised of actin, tropomyosin, and a troponin complex. And the thick filament is then comprised of myosin, both a light chain and a heavy chain. And you can see the light chain has a regulatory component with ATPase activity, as well as a stabilizing component in the alkali light chain. And then it has a heavy chain with a tail, a hinge, and a head. And so what you can see here is much like you could observe in this previous slide. We have the thick filaments, the thin filaments are all arranged here. So these heads are going to be going up towards the thin filaments and interacting with this complex chain and ultrastructure to enable cardiomyocyte shortening and contraction. And so contraction, we generally think of it as being broken down into two major components. One is where there are conformational changes in tropomyosin, and that we consider to have two phases. And then we have the myosin cross-bridge cycling phase, which then has 5 phases to consider. So when we're looking at resting conditions, this tropomyosin complex, where there's not cardiac contraction, there's not cellular depolarization, we have the troponin complex largely blocking a myosin binding site on F-actin. And so you can see here that there's this myosin binding site to the myosin head being down here. Myosin binding site is unavailable because we have this troponin complex on this sort of bead of tropomyosin being blocked by troponin I, or the inhibitory part of troponin I. And so when we get that increase in intracellular calcium secondary to an increase in calcium-induced calcium release during early phases of cellular depolarization, calcium then binds to troponin C, so troponin C being the calcium binding subunit of this complex, and that results in a conformational change, which then makes this myosin binding site available for binding on the F-actin molecule. And so you've got troponin T here, which again is largely a structural protein that enables this interaction with tropomyosin. So there, again, increase in intracellular calcium binding to troponin C, and eliciting this conformational shift and exposing the myosin binding site. So now we've got this handful of steps. We've got this increase in intracellular calcium. We have the exposed actin myosin binding site, and then what happens next? So generally speaking, at the onset of a depolarization, ATP is bound to the myosin head, and so when ATP is bound to that myosin head, we have the dissociation of the actin myosin complex. And so that's important because certainly in cases where you have depletion of ATP, you will not have dissociation of that actin myosin complex to start off with. And then ATP is subsequently hydrolyzed by the ATPase activity in the myosin head, and that causes the myosin heads to return to their resting conformation, or to the cocked state of the myosin head conformation, which is shown here. So you have the hydrolysis of ATP. A new cross bridge then forms in the myosin head binds to a new position on actin. So now you've got this cross-bridge state here. So we go from the release state to the cocked state after the hydrolysis of ATP, moving now to the cross-bridge state. So binding on a new actin head, actin head and myosin binding site on it. And then with the release of that inorganic phosphate from ATP hydrolysis, the myosin head then changes conformation, resulting in a power stroke. And the filaments then slide past one another and shorten. And then ADP is released. And once ADP is released, the filaments remain in that attached state until another ATP molecule comes along and binds to it, resulting in its release, and then the subsequent repeating of this cycle. And so if you sort of think of what this looks like in vivo, the best, the least expensive way I can think of showing it to you is basically taking your arms and putting them like this, and it's the repetitive cross-bridge cycling, which hopefully is easy to see here, that enables the shortening of the cardiomyocytes through this repetitive cross-bridge cycling, ATP hydrolysis, and inorganic phosphate release. Now, when we look at the sarcomere structure and how this all fits together in the context of a sarcomere, you can see, we've got the thick filament in the middle here and thin filaments attached to these at the Z discs here on the end, and then we have a very large protein here. It's somewhat of an elastic protein called titin, which keeps the thick filaments in the appropriate orientation to the thin filaments. And so again, as you can see, these thick filaments are going to progressively pull themselves down this thin filament and enable cross-bridge shortening with the increases in intracellular calcium. Now, when you take a series of these sarcomeres and we line them up, then you have essentially a myofibril, which is not so dissimilar to what is observed, at least structurally, with skeletal muscle. And so, again, the end game here really being the shortening of that muscle with contraction, enabling, instead of just skeletal muscle movement or limb movement, now the shortening of the cardiomyocytes in the ventricular tissue and the ejection of blood enter right out of the aortic valve or out of the pulmonic valve. So when we look now at the two events together, the action potential as well as the force that's being generated here, you can see a couple things from this figure. So one is we have this very rapid depolarization here, along with a sustained calcium-induced calcium release phase. You can see that during the latter portion of the calcium-induced calcium release, that's where we're having this most significant increases in force production here, and so we have this gradual uptick in force, and then subsequently declines during the decay of the action potential, and so again, what you can see is that as we reduce the magnitude of that action potential, then we're subsequently having significant reductions in the magnitude of force that is also being produced here. So again, that calcium phase, how much calcium you get into the cell and subsequently elicits calcium-induced calcium release, is a major determinant of how much force you're actually going to be generating. So now, I think that for today, that's really what we wanted to cover, and so what we've discussed is how there's the initiation of the action potential, certainly initiating initially at the point of the SA node, and how that ultimately results in an action potential that moves through the conduction system and cardiomyocytes within the ventricle. It results in cardiomyocyte depolarization and ultimately cross-bridge cycling after calcium-induced calcium release and ATP-dependent myosin head cycling. And this is really contraction from a cellular perspective. And so subsequently, what we'd like to focus on next, and in our next lecture, is how these electrical and cellular events actually translate into cardiac organ function. So now we're going to start to move from a very cellular and electrical reductionist perspective of how things are functioning in the heart to how these convert and translate into entire organ function. So look forward to the next lecture, and then thank you very much.